Good morning, everyone. Welcome to the 10th lecture of the PNS mobile genomics. As Mohammed mentioned, today we are going to cover another topic other than the previous lectures. We are going to talk about the genomic data sharing, what are the limitations and the challenges, and one of these limitations is the data privacy. How can we overcome this challenge? And then we will talk about one of the rigorous mechanisms to keep the privacy of the genomic data, which is the differential privacy. We will go for the limitation of applying differential privacy directly to the genomic data and how we overcome this and how we mitigate this. So let's get started. I think all of you are familiar with this, but let me briefly summarize some background information about the genomic data, and let's refresh your information. So inside each one and each cell of your body, there is a code, okay? So again, this code is code of instruction for life. We can consider it as the ingredient list recipe for making every living thing. These recipes are uniquely yours. You can consider it as the invisible signature for you. This signature is written in 4 letters, A, C, G, and T. And when we say genome, it is like the fancy word for the entire collection of the DNA. Now, if you remember, Mohammed, like in his lectures, talked a lot about the genome sequencing. So the genome sequencing simply it means the revealing and putting in order the C, G, A, and T. Okay, so if we consider that genome as the instruction manual for you, so when we say gene, so the gene are the individual chapters within that notebook. Now we have gene codes for making eyes, for making hearts, and we all have these genes. And in fact, we are 99.9% genetically identical across all humans, but we have 0.1 variations. So these variations, like between us, makes us unique. So unique in the color of eyes, unique in the height, also like unique in response to medication, unique in predisposition to disease, and so on. So from where you are getting these genomic data. So mainly you are inheriting these genomic data, of course, from your father and mother. So 50% you are getting these data from your father and 50% from your mother. So for each location of that variation, so if we consider these variations, like we call it single nucleotide polymorphisms, or imagine that these are the locations in your DNA. So each location, each SNP have 2 alleles, again, one from the father and one from the mother. Now, for each SNP, these 2 alleles can be like, the alleles itself can be major allele, which we represent here by the capital B, or can be minor allele, which we represent as small letter b. Now, if you inherit both major alleles from your father and mother, then we call it homozygous major, that is SNP, that location in your DNA. And in that case, we give the value of 0, because here you have 0 minor alleles. If you inherit one major from the mother and one minor from the father, or like major from father and minor from mother, then at the end you will have 1 minor allele, and here we call it heterozygous, and we give the value 1. If both are minor alleles, then we call it homozygous minor, and we give the value of 2. Now, what is the probability of having the values of 0, 1, or 2, or if like what are the probabilities of having like homozygous major SNP or heterozygous SNP or homozygous minor SNP? So these are the Mendel's laws. So in Mendel's law, like for example, imagine here we have a father and we have a mother. So the probability, like Mendel's show the probability of having these values. Let's pick an example. Here, if the father has the value 1 or heterozygous, and the mother, for example, like have the value of 0, which homozygous major, then you are 50%. Like the probability of having 0 will be 50%, and the probability of having 1 will be also 50%. Again, here also we have the table if we consider the child and mother. So what are the probabilities for the father alleles or SNPs values? So also you can pick it from here. Okay, so now we need this genetic information, and these genetic information are very helpful for the advancement of the research and also healthcare directions, biological direction, bioinformatics, and so on. Now, we all know that currently we are in the genomic era, and again, the genome holds the promise to help protect your health. And actually, in the United States, the genetic factor are known to play a role of in 9 of 10 leading causes for death. All right. So currently, we have more than 2.5 million sequenced genome around the world, and this number is projected to reach more than 100 million sequenced human genome in the next 3 years. Okay. Especially after the COVID pandemic, many countries decide to study the genomic data in a population scale. They want to leverage the power of genome because the researcher again need large amount of genomic data set that they can leverage to gain better understanding and your genetic data, also the association between the genetic data and phenotypes or disease, let's say. Now, we have again a lot of projects which considering like population, the whole population for sequencing their genomic data, study these genomic data, and like these data is like rich trove of data. You can imagine how much like the power it can give to the scientific advances, right? Now, okay, we are trying to do these projects in a population scale. How about the populations? What is their attitudes toward or donating their DNA, donating their genomic data? So we have several studies report the attitude of public in different countries like the States, Sweden, Japan, Singapore, towards genomic research, and the willingness of the people to donate the genomic samples. So 70 to 90% of the participants in these studies had positive attitudes. They want to donate, but of course they have some concerns. So what's like what the concern or the risk we are talking about here? Here, we are talking about one of the challenges, which is social, not technical. We are talking about the data privacy. According to the sensitive nature of the genomic information, sharing these genomic data set with the sensitive genetic or medical information for individual can be, of course, misused if it lands in the wrong hands. They can do a lot. What are like these risks? For example, if the owner of a genome is identified, then he or she will face like the risk of discrimination by employers or insurance companies. So here we are talking about like this information as financially valuable, all right? And also because, as we know, like the DNA sequences are highly correlated, right? So if one of like the genetic record of an individual is leaked, it's leaked for good. It's not like a mobile password that you if it is released, then you cannot change it. If it is then again leaked, it's leaked for good and it's done. And it's not only about one individual privacy, we are talking about the whole family then. And I think you know the example of Henrietta Lacks, or you can also, Mohammed already mentioned about it. Also, you can read more about it and how the whole family suffer from the privacy risks because of the genomic of one of the family members. Okay, so now we will go to see one of the genomic data examples in which we are not releasing the records. We are not releasing the DNA of individuals. We are releasing summary statistics about the participants. So again, let's recall the genome-wide association study. And these studies, the goal of these studies is to find the position in the human genome that are like, we need to find these positions that are statistically correlated with the phenotype. When we say phenotype, let's say height, let's say color of eyes, or maybe disease like Alzheimer, any disorder we have like cancer and so on. So the GWAS is case control studies. It means that we have cases who have this phenotype or this disease and controls who are healthy or disease-free, all right? Now, what we have from these studies are these tables in a way that we are like for each case and for each control, we are trying to find how many of them have the value 0 at that SNP, how many of them have the 1 value and the 2 value. So we are only counting all of these numbers, and the output of such studies again are statistics. So we are talking about chi-square statistics. We are talking about the p-value because from the p-value, we could know the association between that SNP position and one disease or one phenotype. We are talking also about minor allele frequencies. We want to know the frequency of the minor alleles here. We are talking about sometimes the count. So we have summary statistics at the end. So these data set, again, these studies are considered to be safe, right? We are not showing the genomic record of any participant. We are only showing the statistics. So again, it's considered to be safe until one attack happened, which called RowHammer attack. And like briefly, this attack is like this. They are trying to know if one of, like we have victim or target, they're trying to know if this target is a member or he is in the case group. Because if I could know that, for example, let's say we have a highly sensitive genomic data set. It is about the hepatitis, for example. So if I know that that person or that victim is in the case group, then it's done. Like I could leak his sensitive information. I could know that he has that disease and so. So after that attack happened and they were successful in finding like if one victim inside the case group. And after that attack, the NIH in the United States, the National Institute of Health restricts the access to key results of the GWAS data. So they only give the permission for qualified or trusted researchers or individuals. Okay, so what's the solution here? We need the genomic data sharing. It cannot be like this. I mean, we want the access to be wide access for this genomic data. So for the hope of sharing the genomic data set to gain better understanding of the human genetics, a lot of studies propose different techniques for the genomic data set in private manner. Now, one of the most common and rigorous mechanisms is the differential privacy. So what's the differential privacy? How we can use it for genomic data? What are the limitations? How we can mitigate these limitations? This is what we will know next. Okay, so in general, if we are talking about the genomic data privacy, we need to consider both the computing and also the sharing. So what we consider here in this lecture is the sharing of the genomic data. So let's go again for the differential privacy. So simply, what's the differential privacy? Now, imagine that we have adversary. He wants to query the data set, the GWAS. He is asking, for example, give me the chi-square value or give me the p-value. Now, that adversary will be confused, or he will not be able to distinguish the answer produced is from dataset A or dataset B. Like he said that, okay, when I get that result, I could know that, okay, this target or this victim is inside the dataset. Here, we want to hide the impact of one individual. So imagine like we have a data set and we have a neighbor data set. So both of them are the same. They differ only in one row. So if the adversary sends his query to the data set, he will be confused from where the answer I got. It is returned by this data or the other data. So this is the brief idea of differential privacy. More technical, like let me give you more technical details about it. Then again, imagine that we have two data sets, we call them neighboring data set. These two data sets differ in one row, okay? So based on the differences, we have two types. If they are in the same size, but they differ in the content of one row, we call it bounded differential privacy. If they have, like one of them have one extra row, then we call unbounded differential privacy. Now, again, the main idea here, we want like to have the probability of having an answer from T, and the probability of having an answer from T bar will be only, you know, or like will differ by bounded multiplicative factor we show here, like e to the power epsilon. So simply, what's e to the power epsilon? Here, epsilon as the privacy budget, okay? So based on the sensitivity of the data or of the queries, we choose that epsilon value, all right? Now, if we have like very sensitive, the nature of the data is very sensitive, I want to add like mainly, I'm adding some noise to the results. Like we have, for example, the minor allele frequency results. I will not release it to the like adversary or to any researcher as it is. I will add some noise. So if we have very sensitive data, maybe I will add more noise. If I have less sensitive data, I care more about the accuracy or the utility, then I need to add less noise. So how we can choose the level of noise? It is based on that epsilon, which is the privacy budget. The less the privacy budget, the more privacy we have, and the less utility, of course, we have. So how to achieve differential privacy, how to add the noise? There are different general approaches to achieve differential privacy. One of the most common is adding Laplace, like perturbation mechanism, adding noise based on the Laplace distribution. So the Laplace distribution, again, we have mean and scale. So here we decide the scale based on the query. For example, here we have the query over the T or the query over the T bar. Then we are adding some noise. How to add that noise? It is based on the sensitivity. What is the sensitivity? It is the maximum change between the results. So for example, if you are talking about some query, what's the maximum change in the sum result for both data sets? So this is what we call the global sensitivity. Then at the end, we are adding noise, okay? So we have also other approaches like Gaussian, like exponential, and so. Here, since we are dealing with a numerical data set, so Laplace fits our genomic data sets. The differential privacy currently used by many companies, big companies for collecting the data, for example, like Google for the crowdsourcing data, by Apple for examining the information shared from the customers, also Microsoft Research, and finally like Census company and one of the biggest census company in the United States. All right, let's go now for the differential privacy for genomic data. Can we directly apply it? Can we just query the data set, add that noise, and then release it, and then it's fine, we are safe, it's privacy preserved? Of course, no. We have some limitations if we want to apply the differential privacy for the genomic dataset. What are these limitations? The first problem we have here is the differential privacy don't consider the correlation between the data set. It means in the differential privacy, we assume all the data we have, all the rows we have are independent. But in the genomic data, of course, if we have like family members, so they are really correlated. We know that they are genetically correlated. Then the data we have is correlated. Then because in the differential privacy, there is no assumption about the correlation of the data, this degrades the privacy guarantees of the differential privacy. All right, now we are trying to solve this problem. What are the state-of-the-art studies try to do? Again, they want to, again, increase the privacy levels. But when they try to increase the privacy levels, most of them or many of them are trying to add more noise. We want to add more noise, then we can overcome the limitation. But this is not the perfect solution, because if you are adding more noise and more noise and more noise, this is to poor accuracy, right? What I need from the query result in that case, if I'm adding too much noise, how much utility I have, how much accuracy we have. So we need to find a way to, again, have the privacy guarantees, and also we need a trade-off between the privacy guarantees we have and also the accuracy or utility of the data. Now, again, many of the related work started to find this limitation. The first one to find the limitation of differential privacy is the pufferfish framework. And here they didn't show any mitigation. They just show that we have that limitation for differential privacy. Following the bufferfish, we have bluefish, we have patient differential privacy. They're trying to give some mitigation or perturbation mechanism, but here they consider, like for example, in the bluefish, they have some restriction which cannot be generalized, and also the same for the patient differential privacy. They model it by Gaussian Markov random field, which is also cannot be generalized for any data set. Following them, we have also another works which try to solve these problems and propose mitigation techniques for overcome the challenge or limitation of differential privacy. But again, some of them are adding more noise, then we have less utility. Some of them, for example, and here in the dependent DB framework, they're only considering the pairwise correlation, 1-to-1, 1-to-1. But in the genomic data, we have joint correlation. For example, if I have mother, father, and son, then 3 of them are correlated together. I mean, the son to his father and his mother together, right? So I cannot go for pairwise correlation. We need to consider joint correlation for more accurate data. So what are our contribution in that direction? First of all, we try to find or investigate the feasibility of the attacks, right? So we have 2 types of attacks. The first one, we are trying to know or find or infer the genomic data of one victim or one target. I want to know the DNA. I know that he is in that data set. I know that this guy participated in the data set, but I don't know the value of the system. Is it 0, 1, or 2? So I want to infer that one. We call it attribute inference attack. And what we did in this direction is we try to use the differentially private sum query, also MEF, the minor allele frequency, and the chi-square in a static genomic dataset. What's the static genomic dataset? It is the data which is updated for one time. When we are talking about dynamic, this data can be updated anytime. One good example for dynamic dataset, for example, is the clinical data or the hospital's data, like when we have patients who can join and leave anytime and so. But here we are talking about one-time updated data, which is static dataset, and we use the results from some query, MEF, chi-square, to infer the genomic data of one target or one victim. The other inference attack we can do is the membership. Now I have the DNA, I know the genomic record, but I want to know, is this victim, is this target included in the dataset, included in the GWAS, included in the case group in the GWAS or no? Now we could like infer the genomic data, and we could also infer the membership of one victim, like to know that he is part of the case group and GWAS studies. Now, to mitigate these inference attacks, we propose two countermeasures. The first one is based on working or playing with the Laplace perturbation mechanism. We are trying to change the noise, but we want smart change. I don't want to add too much noise, or I don't want to add less noise. I want to find accurate number for that noise. I want to compute the sensitivity of the query as we have this number of correlated participants in the data set. So in that case, again, we are working in the Laplace perturbation mechanism. The other countermeasure here we have is we are not touching the differential privacy mechanism. We are working on the data set itself. We are trying to hide some SNPs. We can find some sensitive SNPs which can reveal the kinship relationship or the relativeness between that person and that person. This SNP can show the relationship between participant A and participant B. Then I can hide this SNP and so. So we are trying to hide some SNPs, and after that we apply the standard differential privacy without changing anything in the mechanism. Now, in all of these studies we use real-world data sets from the 1,000 genomes project. This 1,000 genomes project. We have genomic data for more than 2,500 participants from different population areas. And also we have two family data sets. The first one is Manuel Corpus family. This one, Manuel Corpus is a scientist. He shared the genomic data of himself and some of his family members for like the improvement or for empowering the scientific research of genomic data and also the biomedical research. We have another family, which is Utah family. We have the data for the parents, the grandparents, and 11 siblings. Okay, so we are using these data sets for our attacks and also for our mitigation. So the first attack we have is the attribute inference attack. What we have in this attack is like this. Again, we consider, we consider that adversary, he is sending a query over data set, he received this, let's say we have some query, he received the answer of that some query, and he knows that the family members, these family members are included in the data set. From where he could know, from some side channel leaks, like let's say social media, maybe it is targeted attack in which that he knows that this family are these family members are participating in that data set. So using this information, like that the family members are included, plus the sum query we have, he could infer. How he could infer, let's go for the algorithm. Again, that adversary generate the query includes the members of the same family. Let's say he can say, give me the sum query of, give me the sum value of SNP A for the people who live in Zürich, for example, Zürich. Then now he knows that the family members are in Zürich. So because he formed the query based on location, or sometimes he add the age, then he will be sure that all the members will be included in the query result, right? Now, in our work, we show that there is dependence between, again, the family member sum and that target or that victim values. Then we could find the dependence equation, and that's the thing that the adversary will use to estimate. How he can estimate? Let's say he will get the sum, and the sum value he got, he get as he received as 6. Now he will apply the coinage change algorithm. What's the coinage change? First, we need to find all the possible partitions. Now for that 6, how many partitions we have for 0, how many partitions we have for 1, how many partitions for 2. Again, so for example here, the second one, we can have 1 with 0 value, 2 with 1 value, and 1 with 2 values, right? Now, based on the participants of included inside the query result, he could find what are the correct partitions and which partitions we need to exclude or to ignore. So for example, that one, the third one, we have partitions for 5, but here the number of participants are 4, then we ignore that. Of course, we have a lot, many other partitions, but here we show like a simple example. Now, after that, he will catch the correct partitions, and he will try to find the permutation for each one of these. Like for example, the 6 can be 1, 2, 2, 1, or 1, 2, 1, 0, for example, based on these 2 partitions, right? Now, the adversary knows that the family members are included. He knows that, okay, we have the father, the mother, 2 sons, for examples, and so do you remember the Mendel's law at the beginning? Then here he will use the Mendel's law to validate the permutation we have. Like, for example, if we have the father and mother like with 1 and 2, it's impossible in the normal ways, or let's say like if we have like the if we go directly to the Mendel's law, it's impossible to have the value 0. Okay, so if the father and mother have 1 or 2, then the son can be again 1 or 2, but he cannot be 0 at all. Okay, in the normal, I mean, ways. All right, so without having any disorder, for example, his genetic or any other things. So based on that, maybe the son will have, for example, the value of 1 or the value of 2 in the same partition for the same, like we have different permutations. So the adversary will choose the high, like the permutation, the value with high probability. Again, he will use Mendel's law. If you remember in Mendel's law, we have probabilities. The son can be 50% 1, the son can be 50% 2. In that case, the son can be 1 or 2 in the same probabilities. Then he can pick one of them, for example. Sometimes they can say, no, the son can have have the probability of 50% of having 1, but 25% of having 2. Then I'm choosing the value of 1 for him. Then, like for more, I mean, like for more family members and more permutations we have, more partitions, he will calculate the probabilities, and then he will try to find the genomic value or the record for that SNP for the vector. Okay, so after that, again, as I mentioned, he will use the Mendel's law. And how we quantify the success of the attack, we compare what's the value the adversary can infer and what's the correct value, the original value we have. Based on that, we can say, okay, he was correct in 90% of them. He has some like estimation errors of, let's say, 70%, 30%, and so. And also we can calculate how many leaked positions. For example, if we are considering 100 SNPs, how many out of these 100 he could infer correctly. So these are the attacks, like the metrics we use. And the key results we have is that the adversary could infer the actual value, the correct value of the targeted SNPs by up to 50%. And we try to mitigate in this work, but in an using empirical way, in which, like, for example, we said, okay, if we are using epsilon with that value, we have like this correctness for the adversary, or we have that amount of error. Let's decrease that epsilon. And somehow, so we did some experiments, and using the empirical results we have, we could find a mitigation mechanism to mitigate these inference attacks, okay, as a countermeasure for these inference attacks. And using our mechanism, we can achieve up to 50% better privacy guarantees. For example, the differential privacy, traditional one can leak that much. We could limit that privacy degradation by 50%. Okay, so you can find all the details, all the algorithms we use, all the results in our papers. It's published in the bioinformatics, and it's also open source. Now, let's move to the second work we have, in which that here we try to do 2 attacks. First, attribute inference attacks. I want to know the DNA, the genomic record. Then I will use that genomic record to find the membership of that target in another dataset. Why we can, or how we can do such thing. Let's imagine again, we have 2 datasets. One is sensitive and one is highly sensitive. So I can query the sensitive data. Let's say it is about phenotype, color of eyes, color of hair. Then I can infer the genomic data, genomic record of one victim. Then I will take that genomic record and I will use it in a highly sensitive data set. Let's say hepatitis. Then I want to know, is that person inside the case group or not? So this is the thing that we succeed to do in that work. We did attribute inference attacks over genomic data set, like we have GWAS and we have any statistical data set. We ask, like, we send the queries for MEF, chi-square statistics, and using the results from these queries, we could infer the target SNP. Again, we use the inferred SNPs in another attack, which is the membership inference attacks. And again, we try to find if that participant or that victim is in the case group. After receiving the MEF query results, we could decide that target is in the case or not. Now, how we can do it in more details? Again, the adversary here could know the MEF for the control. It can be released with the data set. Also, he could know, like, the MEF for the population, the whole population, the whole city here or so. He will query the MEF for the data set, including both cases and controls, right? Because he knows the controls, he can apply the log likelihood ratio. And from using that test, he could know that target is part of the cases or not. He has 2 hypotheses. So he is not, or he is in the case groups. So based on the results you will have after sending many queries, he could infer the membership of that target inside the dataset. What are the key results we have? Then the adversary can reveal up to 40 to 50% more sensitive information if we are talking about the attribute inference attacks. And also he could infer the membership of a target or a victim, and the inference power of the adversary can be high in the membership. After sending some queries, he can infer the membership of that victim. You can find more details about the 2 inference attacks in our paper. It's again presented in the ISMB and published in the bioinformatics, and it's open source as well. Then you can find there all the algorithms we have, examples of what we did and so. Now we are done with the inferences or the attacks we have. Let's go for the mitigation parts. So the first one is, I told you that sometimes we are not trying to play with the Laplace perturbation mechanism or with the DP. We are trying to play with the data set itself. So here we try to selectively hide some SNPs. Imagine here we have data set D. So when the family members join the data set, we are hiding some parts of the SNPs, which can reveal the kinship between those, which can reveal sensitive information between the family members. So the idea here, we want to distort the correlation, then we can use the differential privacy. So now when the adversary try to query the data set, we can add the Laplace perturbation mechanism, the standard differential privacy. We are not increasing, the epsilon value, we're not playing with the sensitivity at all. So we are using the general Laplace perturbation mechanism, and we reveal the response to the adversary. That's why he couldn't, in that way, he will not be able to infer the genetic record for that victim. How we selectively hide? So imagine that we have the manual corpus family data. Then, for example, when the son, which is manual corpus, joined the data, it's fine for us. Like he is joining, then no need to hide. But when his father, for example, joined, then we are trying to find what are the sensitive places, what are the available SNPs to be hidden. So we collect, like we try to collect all of these available. Now, we choose from these sensitive SNPs, some of them to be hidden. All right, this is our current approach we are using. And then when the mother joined the data set, then again, we are trying to find the available SNPs to be hidden. Now we are trying to hide the SNP's position, which is overlapped with the father. In that case, we are increasing the privacy. In a way, we still also have a good utility or a good accuracy. So the key results in this work, what we are trying again, we are trying to mitigate the limitation of differential privacy. We try to overcome the challenge of differential privacy, the privacy degradation. So I want to reach the privacy guarantees of the standard differential privacy. I want to overcome any degradation of the privacy because of the correlation between the data sets. So we could reach that, and we could show that, okay, this data set we have, it looks like that we don't have any dependent tuple inside this data set. You can consider it as independent data set. Now, again, much details about this paper you can find in the archive version. This paper is in the process of publication, so you can also find the code of that paper. It is open source, so you can use it. You can find a demo there and examples and how to run our algorithms. Now, the second mitigation way is to work with the Laplace perturbation mechanism to change the query sensitivity because we have correlations, or to change the epsilon value we have. So in this work, GenShare Model, we call it. So here we are receiving the query from the client, from it can be any researcher, and maybe it will be an adversary, right? So let's say we want to compute the MAF, and the data owner will see or will check how many family members are included in the query result, how many unrelated participants are included in the data in the query result. Based on that, he will make a new computation for the sensitivity of the query, and also based on that, he will try to add more noise because of the correlation, but this noise is accurately computed based on the number of correlated data or family members included in the data set. Then he released that MAF with the noise, but again, this noise is accurately computed based on the number of family members in the query result. So you can find also all the details of this paper in the archive version here. You can find all the results we have, how we theoretically find the equations to compute the sensitivity of the query, and also how we are using it in different cases. If we have this number of unrelated participants, if we have this number of family members, and so on. So the overview of our contributions or our work is like this. We have adversary. He is trying to do attacks in both types, the inference attribute inference attacks and the membership inference attacks. So we show that we can do these attacks. We investigate the feasibility of such attacks, and we are trying to mitigate these attacks. So we have two ways or two countermeasures. The first one is by selectively hide some SNPs, and the other one is by using or again changing the scale in the Laplace perturbation countermeasure. So that's all for today's lecture. If you have any question, I will be happy to answer. Okay, then. Thank you so much for attending. I hope you enjoyed the lecture today and it was useful for all of you. And yeah, thanks again.